Hello, good evening and a happy Wednesday evening to you all. Welcome to the first NHM Live of 2019, coming to you live from the Natural History Museum here in London. My name is Eddie and I am going to be your host for this evening, and do we have a fantastic show lined up for you. We are going to be taking a behind the scenes look at our imaging labs, checking out some of the funnier looking members of our collection, as well as revealing our mystery specimen. Before we get to all of that though, please do not hesitate to get in contact with us however you're watching today, on Twitter, on Facebook or on YouTube. Send in just saying hello, where you're watching from, or any questions you might have for our museum experts. Right now, let's jump over to Josh in the newsroom for the NHM Live News Update.
Run it.
Hello! This month saw China make history when it successfully landed its Chang Air Force spacecraft on the far side of the moon. Touching down on January the 3rd, the lander aims to study the moon's geology and analyse the solar wind. It even managed to germinate the first plant on the moon after a cottonseed it is carrying successfully sprouted before quickly succumbing to the freezing temperatures. But the beginning of the year also marked the first extinction of 2019 with the death of George the Hawaiian tree snail. The 14-year-old snail was the last of his kind after the rest of his species had been eaten into extinction by the invasive rosy tree snail, introduced in 1955. Finally, we can say hello to a new species of velvet spider, named after the late rock star Lou Reed. The tiny spider comes from the coastal hills of southern Spain and lives in burrows underground, and belongs to the genus Louredia, which is named after the lead singer and guitarist of Velvet Underground, and is the first time the group has been found in Europe. Now, back to the studio with Eddie.
Thank you very much, Josh. So we saw in that last story there about the discovery that pterosaurs have feathers. And to dive a little bit deeper to that, we are joined here on the NHM Live sofa by Kieran Miles, one of our paleontologists here at the Natural History Museum. Kieran, thank you so much for joining us.
You're welcome, Eddie.
So let's take a little bit of a deeper dive into this story. We have found out that pterosaurs have feathers. Now, first of all, I think we need to establish something, because people might know that it is quite common knowledge now that dinosaurs do in fact have feathers. Are pterosaurs part of the dinosaur group?
No, pterosaurs are their own unique separate group of prehistoric reptiles. They were the first vertebrates to evolve the ability to fly, and they did this with wings that are constructed in a unique way. They're a sort of stiff, strengthened membrane supported by one hyper long extended finger, mostly. They're considered probably the closest relatives of the dinosaurs. We consider them the sister group to dinosaurs. They probably shared a common ancestor with them sometime in the Triassic. But they're not flying dinosaurs, no. Pigeons, sparrows and crows are flying dinosaurs. Pterosaurs are their own thing.
Fantastic stuff. So this discovery that pterosaurs did in fact have feathers, why is this quite exciting? Why is this so significant?
Yeah, it kind of changes our view of pterosaurs a bit, because we always knew for a long time that they had a sort of fuzzy body coating with the stuff that we've been termed pycnofibers, to kind of distinguish it from the fur that we can see on mammals or the feathers that we see on birds. But this study seems to suggest that this fuzz is in fact true feathers.
And do we have any idea what the sort of function of these feathers for? A lot of like birds today have feathers for sort of like waterproofing or flight. Is it a similar sort of story with our pterosaurs?
Oh, it's highly likely that pterosaurs were endotherms, so probably warm-blooded like us. So this sort of fuzzy body coating probably evolved long before they could fly as a sort of insulation to keep them warm. And we think the same thing actually applies to feathers. They probably evolved first as insulation and then later co-opted to be useful in flight. But pterosaurs didn't need flight feathers, of course, because they had their own sort of membranous wing. Although this particular one, they seem to have had some fibres on their wings, which is unusual even for pterosaurs.
This one is quite an amazing looking creature, isn't it? It's of course a recreation, but it looks, those huge eyes.
Yeah, pterosaurs were quite diverse actually, and this is arguably one of the weirdest types, the anurognathids, the sort of gremlin-faced looking creatures with very large eyes, probably hunting insects. So they've got a few superficial bat-like similarities.
Well, you mentioned a little bit before that pterosaurs are that sort of sister group to the dinosaurs. They're quite related to them. Another group that is closely related to dinosaurs is crocodiles and other sort of crocodilian-related things. Does this discovery that pterosaurs, in fact, had these feathers, does it suggest that maybe crocodiles could have also had feathers?
Potentially. It's not out of the question. Yeah, the big sort of revelation potentially from this paper is that the common ancestor of both dinosaurs and pterosaurs had feathers, and both groups actually inherited them and some lost them. So it's not out of the question actually that that's already pushed the origin of feathers back 80 million years older than we thought. But then it could even go back further than that. And the next line on this family tree, if you like, is the crocodilians. So it's not out of the question that a prehistoric feathered crocodile could be discovered.
So there's still plenty of research to sort of be done there to crack that one. One last one I've got to ask. We've got some beautiful birds around the world today. A lot of them are using their feathers for sort of like bright displays, colours, things like peacocks. Do we have any idea if our pterosaurs had equally sort of bright, lovely colours?
As far as I know, this is the first this study that they were just talking about was the first recorded account of melanosomes in pterosaur fuzz. Melanosomes are the cells that sort of contain the pigments containing the colours. You can sometimes tell from the shape of these what colour the feather would have been. And in these cases, they seem to have been the sort of gingery red-brown rusty colour, which means it kind of makes these even more bat-like than they were before, I think.
So really a lot of similarities there. Well, Kieran, thank you so much for joining us. You're going to stick around in the studio for a little while longer. Brilliant. So all throughout the week on social media, we have been teasing our mystery specimen that we have this week. It's a bit of an odd one. We have been having a few guesses coming through, but no one seems to have got it quite yet. We're going to show you a little tease here. Just a little warning. Anyone with trypophobia might be a little bit afraid of this one, but see if you can work it out. Get those guesses in on social media, and we'll be revealing at the end of the show what it is. Well, certainly a very mysterious specimen there. Kieran, do you have any guesses as to what it might be?
Hard to say. I think it looks a little like some sort of anglerfish.
Some kind of anglerfish. Well, as I say, we will be finding out at the end of the episode, so please do keep getting those guesses in. Sometimes, though, here at the museum, we actually have our own mystery specimens that we need to discover a little bit more about. And sometimes to do that, we will send them to our Imaging and Analysis Centre. We're actually going to take you on a little trip behind the scenes now to discover what we find when we look a bit closer at some of our own mystery specimens.
Hi, I'm Amy. I'm one of the imaging specialists in the museum's Imaging and Analysis Centre. This is where we do a lot of different 2D and 3D imaging techniques and also chemical analysis, and this can all help our researchers find out more about our specimens. Let's have a look around.
Hi, I'm Natasha. I'm the curator of meteorites at the museum. Today I'm using the ichthyologist, which is a petrological microscope, to look at enstatite chondrites. This group of meteorites are really interesting because they have the same bulk composition as the Earth, so they can help us understand what our planet formed from.
Hi, my name's Kate Burton. I am a UCL PhD researcher here at the museum, and I am doing my research into creating 3D models using photogrammetry inside this scanning electron microscope to create accurate images of things that you have to have vision to see normally, but in this case they can be tactilely handled by someone with vision impairment.
This is one of our CT scanners. It uses X-rays to build up a model of the inside of an object in 3 dimensions. Today we're scanning a coral specimen, so the scan will be able to tell us about the internal structure of the coral. It's also really useful for looking at fossils inside rocks and at bones and organs inside an animal specimen.
And Amy joins us now live in the studio. And Amy, you've brought along some imaging equipment with you. What do we have here?
Hi Alison. This is one of our handheld structured light scanners. It's basically a couple of cameras and a projector, and I brought this megalodon tooth to show you how it works. So it's projecting a sort of grid pattern onto the object, and the cameras are looking at how that pattern stretches and deforms as it moves over a three-dimensional surface. The little dots on the foam here are how the system keeps track of where the object and the cameras are relative to each other. And you can see it really starts to build up quite quickly.
It is amazing. I'm surprised at how quick it actually works.
Yeah. So the processing a model like this afterwards takes a good bit more time, but the initial capture can be really quite quick.
And we've got some quite incredible, we've got the colour there, haven't we? How detailed can we go with a scan like this?
So the spatial resolution is about 0.2 of a millimetre. At the moment we've got a point cloud rather than a surface. Each of these points is a XYZ coordinate and an RGB colour value. So as you say, we've got a good visual representation as well as the shape and size of the object.
It's brilliant. So you've scanned one side of it. Would you then turn it over and scan the other side?
Yeah. And we can merge the two together and make an entire object without the base.
Fantastic. So does this work for all of our specimens, or are there certain things that it works better on?
It works very, very well for fossils, rocks, bones, things like that, especially the larger specimens. Where we need finer detail, we would use the laser scanner, which doesn't give us colour, but we get much, much finer spatial detail, and works in the same way that we would do the top and the bottom and merge the two together.
It's absolutely amazing to see it. Why are we doing this? Why do we want to digitise our specimens in this way?
So we can take all the same measurements from a digital model that we could from the real specimen, like in terms of size and width. We can also look at things like volume and cross-sectional area, which you can't do with a real specimen. It's a good insurance in case anything gets damaged or broken, and it's a great way to let other people have access to our specimens from other institutions, a lot safer than sending real ones on loans.
Of course, it's brilliant, and thank you so much for bringing and showing us today.
No problem.
Now we're going to find out a little bit more about your 3D imaging work in a second, but before we do, let's take a look at what technology is helping to reveal about our ancient ancestors.
This bone was studied before. What we had to previous study was a 3D analysis, which allowed us to recognize more details on the modification. We recognized cut marks, percussion damage, and even human tooth mark. But there was a set of modification which was difficult to recognize. Using 3D analysis and comparing with other bone, we thought it was actually an engraving on a human bone, which is the oldest example now known. This also suggests that the cannibalistic act was actually part of a ritualistic cannibalism, and they were not eating each other out of necessity.
Well, we're very luckily joined by Amy here on the NHM Live sofa. Amy, thank you so much for coming down to talk to us. So let's talk a little bit more about some of the projects you've been working on. We'll also, please do get your questions in for either Amy or Kieran, because we do have a chance to answer some of those. One of the really big blockbuster projects that you guys worked on, one of the first project you had, was actually scanning Hope, which is our blue whale which hangs up in the Hintsey Hall. That must have been quite a big challenge for the lab.
That's right, yeah. Not only was that the first major project that we had, the timescale was very, very tight. There were a lot of other specialists who all needed to work on the specimen while it was off display. So we actually only had a window of about 6 weeks to do the scanning work, and we were back and forth with other departments during that as well.
So you say that it took about 6 weeks to scan all of the different specimens. How long did it take to sort of get all of that information and bring it all together for a final product of that model?
The processing afterwards took about another 8 months. That's by far the bigger part of a project like that.
Definitely, absolutely. And that's, you're getting it to such a huge detail, and getting it all together must be really useful. Is it sometimes a bit of a challenge working with some of these specimens? Because we can see in some of the images here, some of the vertebrae and the ribs, they're pretty big and they're quite delicate. Is there ever any challenges working with any of them?
Some of these are a 2 or 3 person lift, so it's not just the time it takes to actually do the scan. You need to arrange people to help you move it and move on to the next one, or to turn it over and do the other side. It all becomes logistically a lot more challenging.
But it's still amazing. Well, we'll be talking about where people can see it. Some of the other projects that you've worked on, another quite famous museum specimen we have here is the Archaeopteryx, which is this amazing fossil specimen that we have. And you were actually sort of using the laser scan on it. What was the sort of goal of this project of laser scanning it?
That's right. So the Archaeopteryx was part of our treasures exhibition that toured to Japan and other places. And the idea initially with the laser scan was to have a really detailed condition report, so that we'd be able to compare that after the tour and check for any damage. But what we realised afterwards, we scanned this and also the counterpart fossil that fit together, that we could digitally do exactly that, which we would never attempt with the real fossils. And we can then use the software to measure how closely those two parts fit together.
So yeah, if we look, it will render in just a second, but we can see we've got the sort of different colour levels, these different temperatures. What are these actually representing in our image here?
So the green part here is showing you that there's almost no difference between the two parts at all. They fit exactly perfectly together. So for most of the fossil, and especially on these delicate feather areas, you can see it's a really perfect fit. Where we've got bigger gaps here, anywhere up to 3-4 millimeters, is where the fossil preparators have actually removed matrix material away from the fossil bones, and that's where we've got bigger gaps.
And this is quite significant, isn't it? Because this really proves that this is real. Because am I right in thinking there was actually a belief that this wasn't a real fossil when it was first discovered?
That was alleged, yes. I think more recently than when it was first discovered, and a lot of people in the museum were not happy about that, and there's been a lot of different analyses done which have all completely disproved that idea. But this is just one more that helps us show that it's the real thing.
So, because Kieran, we were mentioning a little bit before about sort of birds and dinosaurs. The Archaeopteryx is kind of this first link that birds and dinosaurs were related. Is that right?
Yeah, at the time it was discovered, it was considered the first bird. It's now unusual more in the fact that it's probably the earliest winged dinosaur, I guess. If we'd found it these days, we'd think it's another feathered dinosaur. But yes, it was the first real inclination that dinosaurs and birds are really closely related.
Fantastic. We've got a question coming through from Catherine on Facebook asking, do we use photogrammetry on specimens?
Sometimes, more often in the field. The structured light scanners don't work if it's too sunny. So that's one time that I have needed to go back to using photogrammetry, is working on fossils and fieldwork sites.
Fantastic. So, Amy, we've been talking about these wonderful models that have been created. Is there any way that the public can access and have a look at these models?
Yeah, so we have a Sketchfab page. You can go on there. There's 80 some models up already, and we're adding to it all the time. You can interact with all of them in 3D or in VR if you have that, and most of them are downloadable as well.
Fantastic. So that means people, if they have access to, can download and 3D print them or something like that.
Yeah, absolutely.
Incredible stuff. So it's a really amazing way to just share all this information. People all across the world. You don't have to be at the museum to do it. Wonderful. Well, thank you so much for answering those questions there. We may get a few more questions in. For now, we've been looking at some of our really amazing specimens, things like Hope, our blue whale, and the Archaeopteryx. But we've got 80 million specimens here in our museum collections, and not all of them can be quite as beautiful. Some of them are slightly weirder looking. So let's take a little look behind the scenes and find some of our funnier looking specimens.
Fangs like a vampire, skin like jelly, killer smile full of sharp teeth, and one with an angle poised lamp on its head. These are the sort of facial features that I like to look for in a fish. I'm Ollie Crimmen, Senior Fish Curator here at the Natural History Museum. I joined the museum in 1973, and over the years I've developed a bit of a passion for fishes with funny faces. I think we anthropomorphise, we put human characters on animals, but fish are a bit of a challenge that way, and their lifestyles make their faces very interesting. For example, the rocksucker clingfish has a ferocious looking face, but those massive teeth are for levering limpets off rocks. Another favourite of mine is the burrowing eel goby Tenoides. With massive fangs, tiny eyes, it moves through its burrow, catching food with those teeth. The blobfish is famously ugly. In fact, it's the mascot of the Ugly Animal Preservation Society. However, it's always photographed out of water, and that jelly-like skin actually helps it survive the enormous pressure at which it lives in the deep ocean. We normally only see the tail of the monkfish on our dinner plates, but the head is broad and the mouth is full of teeth, and there's a lure on the head. It's an anglerfish. It lives in fairly shallow water. Perhaps the strangest of all is the football fish, which is like a globe with a face on the front of it, a huge mouthful of teeth, but that's all invisible because it lives in the dark. It too is an anglerfish, and its luminous lure on its head brings prey within reach.
Well, some pretty unforgettable faces there, I'm sure you will agree. Just very quickly, Amy, Kieran, are there any sort of funny looking faces that you've seen in the collection that you can remember?
Funnier than the pterosaur we saw earlier.
Always quite a funny looking one.
Yeah, in the fossil fish collection next year, we've got some fish that might not be considered what you call conventionally attractive. There's a lovely one called Xiphactinus, which has a sort of bulldog-faced, pretty weird-looking thing.
That sounds like a pretty weird-looking one. Brilliant stuff. Well, we're actually coming up right to the end of the show now, but just before we finish off, there is time to reveal our mystery specimen. Now, we've been getting some brilliant guesses coming through on social media. The most popular guess by far was the naked mole rat. Unfortunately, not right, but a really good guess. A few people went for snail, some for salamander as well. We had one guess for pangolin and one for coral, but there is actually, we did get a guess. First guess right was John on Periscope. But before we reveal, let's find out what it is. And just very quickly, should mention again, if you've got that trypophobia, you might want to give this one a miss. Let's find out what our mystery specimen was. There we have it. It was the Suriname toad. So very well done. So that was John from Periscope and also Caden from Twitter guessed correctly. Suriname toad, very strange creature. It does have those babies that the babies that are kept in its back that then grow out and swim away. Also, interestingly, it doesn't make a croaking noise, it makes a clicking noise using a bone in its throat. So they are a very strange creature indeed. But unfortunately, that is all we have time for on NHM Live this month. Thank you so much for joining us. If you do have any more questions, keep those coming in and we will try to answer a few after the show. And please join us again on the 20th of February for our next show. For now, we'd just like to say thank you very much for watching and have a wonderful evening.